Nature, where bacteria that might be growing in a biofilm are being infected by viruses, and when the virus injects its DNA into the cell can acquire a small piece of that foreign DNA into the CRISPR locus, a place in the genome that consists of repeated sequences that flank these integrated sequences from viruses that are called spacers. And the cell then makes a copy of that integrated sequence in the form of RNA, which is then processed into individual units that include a sequence derived from a virus. Those RNAs combine with a second type of RNA called tracer and a protein called Cas9 to form a surveillance complex, an RNA-guided protein that can search the cell looking for sequences that match the sequence of the guide RNA. And when a match occurs, the CRISPR-Cas protein, Cas9, is able to cut double-stranded DNA, triggering destruction of the DNA in a bacterial cell. This slide shows the 3 steps in CRISPR-acquired immunity that include adaptation, expression, and interference. What is the function of the protein called CRISPR-Cas9? This is a protein that uses a CRISPR RNA molecule to direct it to a sequence of DNA that matches the CRISPR RNA sequence. And it also requires a second RNA molecule called tracer that provides an interaction with the CRISPR RNA required for assembly with Cas9. And so together, these 2 RNAs guide Cas9 to DNA sequences where the protein Cas9 is able to make a cut in the double helix of DNA. It shows, again, the guide RNA in orange, the DNA strands are in blue and magenta, and you can see how the DNA is held open by the Cas9 protein and allowed to interact with the cleavage sites in the enzyme for precise double-stranded cutting of both strands of the DNA. And then in this image, you can see in green one of the cutting parts of the enzyme swinging into position so they can actually catalyze the chemistry required for DNA strand break. Genome editing extends across all of biology. It can be used for fundamental research, but also for exciting applications in public health, in agriculture, and in biomedicine that causes sickle cell anemia and can now be corrected using CRISPR-Cas9 technology. And this is no longer a potential opportunity, but it's actually been realized by using CRISPR-Cas9 to correct the disease-causing mutation in a patient and to show that this technology is safe and effective for this kind of treatment of genetic disease.